something that consists of atoms joined by bonds, and there are different types of bonds. So first of all, we can talk about covalent bonds. These result from different atoms sharing electrons with each other. So in this example, we've got oxygen atoms. Each of these oxygen atoms has 6 electrons, but in order to be stable, it wants 8 electrons in its outer shell. In order to achieve this, oxygen atoms share their electrons, forming a double covalent bond. Another type of bond is an ionic bond, and this results from the transfer of electrons. So in this example, we've got sodium and chlorine. So sodium has 1 electron in its outer shell and therefore wants to lose this in order to gain 8 in its outer shell. And chlorine has 7 and therefore wants to gain an electron. So what can happen is that the sodium can transfer its electron to the chlorine. Because of this, sodium will gain a positive charge and chlorine becomes a chloride ion with a negative charge. Because positives and negatives attract, this results in the formation of an ionic bond. Lastly, we've got a type of intermolecular bond called a hydrogen bond, and these are an example of an intermolecular bond which occurs between hydrogen and a highly electronegative atom such as fluorine, oxygen or nitrogen. So for this example, we'll look at water, and water is a polar molecule. This is because oxygen has a greater tendency to attract electrons than hydrogen. And because of this, the electrons are further towards the oxygen end of the molecule than the hydrogen end. This results in something called a polar molecule, because one part of the molecule is negative and another part is positive. So then if we bring in another water molecule, the slightly negative part of that water molecule will be attracted to the slightly positive part of the first water molecule. And the attraction between these, which is called electrostatic attraction, is a hydrogen bond. These are quite weak on their own, but if many of them form together, accumulatively they become very strong. So let's now talk about polymerisation. So polymerisation is the process in which monomers join up to form a polymer, and these monomers are going to be joined by covalent bonds. Usually these bonds form as a result of a condensation reaction, and a condensation reaction is something we're going to talk about a bit more later on, but it involves a water molecule being eliminated in order to allow for a new bond to form. So one example of polymers we need to know are polypeptides, also known as proteins. The monomer for polypeptides is the amino acid, which I've shown here. So when 2 amino acids join together, we get a condensation reaction. As you can see here, when these 2 amino acids come together, one of them can lose a hydroxyl group and another one can lose a hydrogen atom. This is going to produce water and allow a new bond to form. So I've shown this here. This forms the new bond, which we call our peptide bond, eliminating our water molecule and producing a dipeptide, which is simply 2 amino acids bonded together. So I've shown here the peptide bond. The elimination of the water simply allowed this to occur, because each atom can only form a certain number of bonds. Another type of polymer we need to know are polysaccharides. The monomer for polysaccharides are monosaccharides, and I've written the general formula for monosaccharides here. So the different types of monosaccharide we can have are glucose, fructose and galactose, and I've shown 2 isomers of glucose here, alpha glucose and beta glucose. You need to know the structure of both of these. The only difference between them is that the hydrogen and hydroxyl groups are basically switched around. Because of this, they have exactly the same chemical formula, and therefore they're both glucose, but they have a slightly different arrangement of atoms, and they're therefore called isomers, which means they have the same chemical formula but a different structure, and therefore have slightly different properties. So our monosaccharides can also undergo a condensation reaction to form a disaccharide, which is simply 2 monosaccharides joined together. In this case, the water is lost from the 2 hydroxyl groups on our monosaccharides. And as you can see, a water molecule can be made up from. What we get formed instead is called a glycosidic bond, which is our oxygen bridge you can see here. So this reaction shown here is alpha glucose plus alpha glucose, which forms the disaccharide maltose. When many monosaccharides join together, then it becomes a polysaccharide, but they're still joined by glycosidic bonds. And this is our glycosidic bond shown with more of the atoms shown. So our structural formulas over here use a corner to represent a carbon atom, but if we write it out, we can see that our carbon is forming the 4 bonds that it should, including one with our oxygen bridge, which forms the glycosidic bond. So these are all of our monosaccharides. When glucose and glucose react, we form maltose. When glucose and fructose react, we form the disaccharide sucrose, which is table sugar. And when glucose and galactose react, we form lactose. Because the glycosidic bond only forms when water is eliminated, which is the condensation reaction, it follows that the bond can be broken by a hydrolysis reaction. Hydrolysis simply means adding water to the glycosidic bond in order to cause it to break. So we can see this in this diagram over here. What we're doing is we're adding our water molecule back in, and because our carbon can only form 4 bonds at any one time, this means that this oxygen bridge glycosidic bond must break in order to allow the water to be incorporated again. So you might have noticed that all of the molecules we've talked about so far have incorporated carbon, and the reason for this is that carbon is capable of forming 4 bonds, which means it's able to form these really varied structures, which means that it's able to form the organic molecules which form the basis of life. So you might have heard about a carbon-carbon backbone before. And this is because carbon atoms are capable of forming 4 bonds. So what we can have is we can have a carbon chain, and attached to this we can still have other chemicals. So maybe we'd have a nitrogen here and a hydrogen over here, and we could have an entirely different atom bonded over here. Because of this, we can form really varied structures whilst maintaining this carbon chain. So we can often say that life is based around carbon. It's also capable and will very readily form other bonds with carbon atoms. So molecules that incorporate carbon in this way are known as organic molecules. So we're now going to look at proteins in a bit more detail. So we know it's a polymer consisting of amino acids joined by our peptide bonds. There are 20 amino acids that naturally occur in proteins found in organisms. And these amino acids can be joined up in any order in order to produce the really diverse array of proteins that we know. So this is the structure of an amino acid in a bit more detail. As you can see, it's made up of carbon, hydrogen, nitrogen and oxygen, as well as a variable chemical group which can contain a whole host of different atoms such as sulfur or even just hydrogen. So this part is called our amino group. This is our basic part, and a similar word for basic is alkaline. If something is basic, it means it's able to form hydroxide ions, which are our OH ions. In order to do this, if the amino acid is in the presence of water, what happens is, if we know the structure of water, we've got 2 hydrogen atoms bonded to oxygen. The amino group is capable of attracting one of these hydrogen ions away and forming a bond. This leaves us with our hydroxide ion, and you can see this here. So our amino group now gets this chemical formula because it's gained a hydrogen ion and therefore has an overall positive charge, as well as forming a hydroxide ion, therefore making it basic. Another part of our amino acid is called the carboxyl group, and this is the acidic part of the amino acid. If something's acidic, it's capable of forming hydrogen ions. In this case, the carboxyl group can simply lose a hydrogen ion, and this is shown here. So again, we've got our now negative chemical formula, as well as a hydrogen ion that's been released, and it's negative because it's lost its positive hydrogen ion. Lastly, we have our R group, and this has a variable composition. And what is the R group determines what type of amino acid it is. So for example, if it's just got hydrogen, the amino acid is called glycine. This represents serine. And there's a whole variety of R groups, which is what produces our 20 amino acids that naturally occur. So as we've talked about, an amino acid plus an amino acid forms a dipeptide, and this is a condensation reaction. And in the same way as polysaccharides, the peptide bond can also be broken by hydrolysis, which is the addition of water. So we need to know a bit more about the structure of proteins. So we can break down the structure of proteins into 4 main levels. So our first one is our primary structure, and this is simply the sequence of our different amino acids. So we've got 20 different amino acids that we can build our proteins from, and these can be joined up by peptide bonds in any order. The order in which they're joined in is coded for by DNA, and this is essentially how DNA controls all of our cellular activities. And therefore DNA ultimately determines the entire structure of proteins, and this is because it determines the sequence. So this is my example of it. The different colours represent different amino acids, and obviously we've only got 4 different colours here, which isn't totally realistic, but it will do. So remember that each of these bonds here represents a peptide bond, and each circle is an amino acid. Next up, we have the secondary structure, and this is the first level of folding that occurs for our protein. So we've got this sequence, but we've got some exposed functional groups on our protein, and when we're talking about the secondary structure, we're talking about the groups exposed on our peptide bond. So in a similar way to if you had loads of magnets like glued together, part of one magnet will be attracted to a part of another magnet. So in the same way, part of one amino acid can be attracted to a part of another amino acid. And when we're talking about the secondary structure, we're just talking about the groups in our peptide bond. So in the same way as we talked about water earlier, the oxygen of the peptide bond is slightly negative because it attracts electrons. And the hydrogen of the peptide bond is slightly positive. So in reality, the hydrogen might be like on that side of the peptide bond. Obviously, this is just a 2D representation. So because each of these peptide bonds has these exposed pairs, so let's say we have an oxygen that side, a hydrogen that side, and an oxygen that side, and a hydrogen that side, etc. These can be attracted to each other, so the protein can sort of fold up a bit, and it folds up into 2 main structures. These are called alpha helices or beta pleated sheets, and the shape it forms depends on the orientation of these peptide bonds. So this is what the structure might look like if it formed a beta pleated sheet. So I talked about this earlier, but this is basically showing what's happening. So yes, we've got a hydrogen bond forming between the oxygen of one peptide bond and the hydrogen of another peptide bond. And as I said, it occurs due to the slightly negative charge in oxygen and the slightly positive charge in the hydrogen, forming the hydrogen bond, which we talked about right at the beginning of the video. So this is showing what an alpha helix looks like, which is this one, and a beta pleated sheet. So our next layer in terms of structure is the tertiary structure, and this is further folding into its ultimate 3D structure. So I've elongated the chain a bit more to show what actually happens. So there are 3 types of bond which allows this tertiary structure to form. These are disulfide bridges, which are simply covalent bonds between 2 sulfur atoms, ionic bonds, and hydrogen bonds. And these hydrogen bonds will be in a different location, so the hydrogen bonds will be involving the variable R group rather than the peptide bond. So the disulfide bridges are the strongest type of bond making up the tertiary structure. Ionic bonds are slightly weaker, and hydrogen bonds are definitely the weakest. But when many hydrogen bonds form together, collectively they can be very strong. So our quaternary structure is our final level of folding of our protein. And this is where multiple polypeptides join together, and they can also join with prosthetic groups, and prosthetic groups are non-protein groups. So if we're talking about hemoglobin, for example, this consists of polypeptide chains as well as iron. So iron is our prosthetic group in this case. So the shape of proteins can be split into 2 main groups. So we can have globular proteins, which are sort of rounded more like this one, and these are for metabolic functions. So a key example would be enzymes. are formed from globular proteins. We can also have fibrous proteins, and these tend to be more like rope, like they look more like rope, and these are used for structural functions. So an example of that would be keratin, which is the protein that makes up your hair. So we also need to know how to determine if a protein is present within a sample, and for this we use a burette test. The burette test tests for the presence of peptide bonds. So first of all, we're going to place the sample in the test tube. We're then going to add an equal volume of sodium hydroxide, making sure it's at room temperature. We're then going to add a few drops of 0.05% copper sulfate, and we're then going to mix this up. So if it turns purple, it indicates the presence of peptide bonds. Otherwise, if there aren't peptide bonds present, it will remain blue, and it remains blue due to the copper sulfate that we're adding. So now we're going to move on to carbohydrates. So we've talked before, the monomer is a monosaccharide, and here they are again. So we have fructose, glucose and galactose, and these can be joined up. So monosaccharides have the general formula CH2ON, which means that the ratio of hydrogen to carbon and oxygen is 2. So for every 1 molecule of carbon, there's going to be 2 molecules of hydrogen and 1 molecule of oxygen. And N can be any number between 3 and 7. So we can have between 3 and 7 carbon atoms and between 6 and 14 hydrogen atoms. So our monosaccharides are sweet tasting and soluble. So the most common example is glucose, which has the formula C6H12O6. And this is a hexose sugar because it contains 6 carbon atoms. And I've talked before about how we have 2 different isomers due to the different arrangement of atoms, which is alpha glucose and beta glucose. So there are 2 types of sugar. There's reducing sugars, and these are capable of carrying out reduction. Reduction is the gain of electrons. So reducing sugar are therefore capable of donating electrons to another chemical. All monosaccharides are reducing sugars, and some disaccharides, such as maltose, are also reducing sugars. So we have a test in order to detect the presence of reducing sugars, and we do this by using Benedict's reagent. So Benedict's reagent is copper 2 sulfate, as well as sodium carbonate, which makes the solution alkaline, and also sodium citrate. The presence of the carbonyl group, which is a carbon double bonded to oxygen, is what makes something a reducing sugar, and this is because this group is capable of donating electrons. Only certain disaccharides have been joined up in such a way that this carbonyl group is actually intact and therefore still capable of donating electrons to other chemicals. So we need to know the method for this. First of all, we're going to add 2 centimetres cubed of our possible carbohydrate solution to a test tube. If our carbohydrate's in solid form, we're first going to have to grind it up in water. We're then going to add an equal volume of Benedict's reagent, which remember is predominantly made up of our copper 2 sulfate. We're then going to heat the mixture in a gently boiling hot water bath for about 5 minutes. So if the reducing sugar is present, the solution turns from blue to orange-brown. So initially it was blue due to the presence of the copper 2 sulfate, but it turns orangey-brown because a red insoluble precipitate of copper 1 oxide is formed. If it's not, the solution will remain blue, and this means that a reducing sugar can't be present. On the other hand, non-reducing sugars are not capable of reducing another chemical, and this is because they have no carbons double bonded to oxygen which are capable of donating electrons. So examples of these are other disaccharides such as sucrose. We therefore need a slightly different test in order to determine if a non-reducing sugar is present. In the same way, if it's a solid, we must first grind it up in water. We're going to then carry out the Benedict's test to determine if a reducing sugar isn't present. If it remains blue, that means a reducing sugar is not present, but a non-reducing sugar may be present, and therefore we're going to continue to the next step. The next step involves trying to hydrolyse our non-reducing sugar, breaking it down into our monosaccharides, because remember that all of our monosaccharides are reducing sugars. In order to do this, we're going to add 2 centimetres cubed of our sample to 2 centimetres cubed of hydrochloric acid. We're then going to place it in gently boiling hot water for about 5 minutes. We then must neutralise our hydrochloric acid because acidic conditions prevent the Benedict's reagent from working. The Benedict's reagent must actually be alkaline. So in order to do this, we're going to add sodium hydrogen carbonate and test with a pH indicator to ensure the solution is alkaline after doing this. If it's not, we just simply have to add more. We're then going to have to retest this new solution using Benedict's reagent. If a non-reducing sugar is present, the solution will now turn orange-brown. If it doesn't, then neither a reducing nor a non-reducing sugar must be present. And I've summed up the reducing sugar test, the Benedict's reagent test, just here again. So our polysaccharides consist of many monosaccharides, and one of the important features of them are that they are insoluble. This is really important for storage because firstly it won't affect the water potential in cells, so it's not going to affect the flow of water into and out of the cells and therefore won't cause the cell to shrink and shrivel or on the other hand to burst. And also it means that it can remain within the cell. If it's soluble, it will dissolve in water and it could easily diffuse out. One of our main polysaccharides is starch, and this is a storage form of glucose which is used in plants. It's predominantly found as granules or grain in plant cell cytoplasm or within organelles such as chloroplasts. Our starch is composed of alpha-glucose monomers, and we get two types of bonds forming between the alpha-glucose monomers, which are 1,4-glycosidic bonds and 1,6-glycosidic bonds. So I've got a diagram showing this here. Our 1,4-glycosidic bonds are the bonds which form this straight chain, and the 1,4 refers to the location of where this bond forms within the glucose molecule. We can also get these 1,6-glycosidic bonds, and the formation of these 1,6-glycosidic bonds forms branches within our starch. So starch is particularly numerous in plant storage organs such as tubers, which are like potatoes, and also in the seeds, because in all of these we need a large amount of energy stored. The reason we have these 1,6-glycosidic bonds forming branch chains is that when they're branched, we can actually obtain the glucose and therefore use this glucose for respiration much quicker. And this is because there are more ends exposed, and since it's the ends that enzymes can work on, we can get more enzymes acting on the starch molecule simultaneously, and therefore more glucose monomers broken off at any one time. So some important features of it are that it's insoluble and large. This means it's not going to affect cell water potential, and as I said, therefore we're not going to draw water into the cells by osmosis. And also because it's large, it's not going to be able to diffuse out because it can't move across the phospholipid bilayer. The fact that our starch is actually wound up into a helical shape also makes it very compact, and therefore we can store a lot of energy in a small amount of space. It can also be hydrolyzed into alpha-glucose, which is our ideal respiratory substrate, so it can be used to directly release energy from respiration. So we can detect the presence of starch using the iodine test. So what we do is add 2 centimeters cubed of our possible starch solution into a test tube, and then we add 2 drops of iodine solution to this. Iodine is yellow. We then shake and stir it, and if starch is present, the yellow iodine turns into a blue-black solution. Lastly, we have glycogen. And glycogen is a storage form of glucose in both animal and bacterial cells. Glycogen is usually found as granules within liver and muscle cells. And we'll hear about this later on in the topics of homeostasis. So, glycogen chains are shorter and more highly branched than starch. And this is related to the fact that animals have a higher respiratory rate than plants. Because of this, and this is because they need more energy because they're more active. And because of this, they're going to therefore need a greater supply of glucose. By being shorter and more highly branched, it means that there's more sites that enzymes can act on in order to hydrolyze the glycogen to release the glucose. So again, it allows for more rapid release of this glucose. And this is a representation of the structure of glycogen. So as you can see, we've still got our 1,6-glycosidic bonds and also our 1,4-glycosidic bonds. And these form straight chains with branches, which then, in a similar way to starch, wind up into a helix shape. So again, glycogen's insoluble. That's again so it doesn't affect water potential and can't diffuse out of cells. It's also compact because it's wound up. It's more branched than starch. So lastly we have cellulose. And cellulose is a structural polysaccharide which is used to make the cell walls of plant cells. This is made up of beta-glucose monomers, unlike our storage polysaccharides which were made up of alpha-glucose monomers. And because of this difference, it results in a different structure, and therefore it's adapted for its different use. So the beta-glucose monomers result in the formation of straight, parallel chains with no branches, and this is due to the way in which our glycosidic bonds form. So this shows the structure of it. We've got our straight chains of beta-glucose monomers, which are joined by our glycosidic bonds. Now between our chains, we actually have hydrogen bonds. So these hydrogen bonds between our beta-glucose monomers result in the parallel chains sort of stacking together, resulting in a very strong structure overall. So the formation of hydrogen bonds between our cellulose, individual cellulose molecules here, results in the formation of microfibrils. We then get further hydrogen bonds between each of these microfibrils, which forms a macrofibril and eventually the whole cellulose fibre. Because of the sheer number of hydrogen bonds, this produces a really strong structural polysaccharide. And another important feature is that it's insoluble, so it's not going to dissolve away in the presence of water. So the last main topic we're going to focus on is lipids. So lipids are slightly different because they're not polymers. The two main types we're focusing on are triglycerides and phospholipids. Lipids consist of the elements carbon, hydrogen and oxygen. They're insoluble in water, but actually soluble in organic solvents such as alcohol or acetone. And this is due to the fact that lipids are very hydrophobic because they don't really have any regions of charge. So they can't really interact with water because of this, but they can interact with other non-polar substances such as our organic solvents. Lastly, the proportion of oxygen compared to carbon and hydrogen is smaller than in carbohydrates, and we'll come to why this is important in a minute. So first of all, we'll talk about triglycerides. So triglycerides consist of one glycerol molecule bonded to 3 fatty acid molecules. This is a condensation reaction, so again water is released, and it forms an ester bond between the glycerol and the fatty acid. So we end up with 3 ester bonds forming. The glycerol molecule is constant, so this doesn't change depending on the triglyceride. The fatty acids on the other hand do change. So fatty acids can be saturated, which means they contain no carbon-carbon double bonds, or unsaturated, which means they do contain carbon-carbon double bonds. Also, the length of the fatty acid chain can also change. So this is another representation of our triglycerides. This part represents the glycerol molecule and this part represents the fatty acids. As you can see, a condensation reaction occurs here, resulting in the formation of our ester bond, highlighted here. So as I said, unsaturated have carbon-carbon double bonds. We can have monounsaturated fatty acids, which just have a single carbon-carbon double bond, or polyunsaturated, which have multiple double bonds. This is important because when there are double bonds, a kink forms within our fatty acid chain, which is highlighted here. This is then going to alter the properties of triglycerides, because when there's a kink in the fatty acid chain, it pushes the fatty acid chains further apart and weakens any intermolecular forces between them. This alters the properties of our triglycerides. So if we think about oils compared to fats like butter, oils contain many carbon-carbon double bonds. As a result of this, we're going to have lots of kinks within our fatty acid structure. Because of this, each triglyceride molecule can't get so close together. This makes it less dense and also means that it has a lower boiling point. Because of this, it's going to be a liquid at room temperature rather than a solid. When there are no double bonds, each fatty acid can pack closer together, and therefore each triglyceride molecule can also pack closer together. The intermolecular bonds between this are going to be much stronger, resulting in a higher boiling point. And because of this, these triglycerides, such as butter, are going to be solid at room temperature. Likewise, within our actual cells, the structure of the fatty acid is also going to change the properties. So the fact that it has a high ratio of carbon and hydrogen to oxygen means that triglycerides are very high energy. This is because our carbon-hydrogen bonds release a lot of energy when they're broken, whereas our carbon-oxygen bonds release far less energy. This means triglycerides can pack much more energy into a certain molecule. They're also insoluble. Again, this means they're not going to diffuse out of the cells and won't affect water potential, so they're good for storing energy. And they also have a low mass to energy ratio. This is really useful because it means we can store a lot of energy with a very little mass. Obviously, if we had to carry around really heavy molecules to store the energy that we require, it would almost defeat the purpose, because you'll need to spend more energy to carry around the really heavy molecules in order to obtain that energy. Lastly, triglycerides can act as a store of water. This is because when they're oxidised, for example in respiration, because they have a high ratio of hydrogen compared to oxygen atoms, they're able to release a lot of water. Organisms that live in deserts are able to use this water because obviously water is in very short supply. So the structure of phospholipids is slightly different. Basically, one of the fatty acids is replaced by a phosphate group which also bonds to the glycerol molecule. This results in both hydrophobic and hydrophilic ends, which is known as a polar molecule. So our fatty acids are unable to interact with water because these are totally non-polar, and therefore they're called hydrophobic. On the other hand, our phosphate group is charged and is therefore able to interact with water. We therefore call this hydrophilic. So the structure can also be represented in this way, with the phosphate and glycerol forming our hydrophilic head and our two fatty acid chains forming the hydrophobic tails. So phospholipids are able to form a phospholipid bilayer due to these hydrophobic and hydrophilic ends. So if we think about our cell, with this representing the cell membrane, we have aqueous conditions both outside the cell and inside the cell. Aqueous means that it's a solution involving water. So we know that our hydrophobic fatty acid tails can't interact with water, but our hydrophilic phosphate head can. Because of this, we get this structure forming, which essentially shields our fatty acids from the water since they can't interact with it. So from this structure you can see, even though we have water outside the cell and inside the cell, the phosphate head completely shield the fatty acid tails from it. So the formation of this phospholipid bilayer is essentially just due to the properties of phospholipids and the way that they interact when exposed to water in this way. They're also important in cell membranes because we can get the formation of glycolipids, in which carbohydrate chains attach to the phosphate head in some way. These are really important as receptors in our cells, which allows for cell communication. So lastly, we need to know how to test for the presence of lipids, and we do this using the emulsion test. So first of all, we're just going to ensure our test tube is dry and grease-free. We're then going to add 2 centimetres cubed of a suspected lipid solution. To that, we add 5 centimetres cubed of ethanol and shake it until the lipid is dissolved. This works because lipids are soluble in organic solvents such as alcohol, and ethanol is obviously an example of that. So if it dissolves, that suggests it's a lipid. We're now going to add 5 centimetres cubed of water and shake it. Because lipids are insoluble in water, if a lipid is present, we get the formation of a cloudy white emulsion. If it's not a lipid, it will be soluble in water too, and therefore the solution will remain clear. So a positive result for the presence of a lipid when we carry out the emulsion test looks like this picture.